Now we are going to start about lipids. So all of you know what are lipids. Lipids are hydrophobic molecules. They are also called as water insoluble molecules. And if you see, mostly lipids are called as fatty acids or esters of fatty acids. Now these lipids are soluble in nonpolar solvents, which are going to be petroleum, benzene, chloroform. Now the general functions of lipids. Lipids act as an energy source. Lipids provide main storage of energy in the adipocytes, and these lipids, they are also functioning as hormones. If you see here, lipids are identified as steroid hormones. These steroid hormones act as intracellular messengers, and there are vitamins which are also classified as lipids, where the lipid soluble vitamins are called as A, E, D and K. So these lipids, they are going to be the carrier of lipid soluble vitamins. Now these lipids, they also help in shock absorption and insulation properties. Now to classify lipids, there are three main types of lipids: simple lipids, compound lipids and derived lipids. So simple lipids, they are called as fats and oils. Compound lipids are going to be called as phospholipids, which are classified with glycerophospholipids, sphingophospholipids. Then you have glycolipids and lipoproteins. Now compound lipids, next to the compound lipids you have derived lipids. So these lipids are derived from both simple and compound lipids, which is acted upon by hydrolysis. So when compound lipids and simple lipids are hydrolyzed, they produce the lipids which are called as derived lipids. Now coming to the basic structure of fatty acids. Fatty acids are called as long straight chain carboxylic acids. So these long straight chain carboxylic acids, they have no branching, and they are of two types of fatty acids: saturated and unsaturated. So saturated, they have double bonds within, they do not have double bonds within the structure, whereas unsaturated contain double bonds in their structure. Okay, so that is about fatty acids. Now coming to the classification of fatty acids, they are classified into two types based on the chain length. Chain length means number of carbon atoms in the structure. So there are three types of fatty acids: short chain up to 4 carbons, and medium chain up to 10 carbon, and long chain fatty acids up to 26 carbon atoms. Now coming to the nomenclature, all fatty acids are called as with their suffix which will end up as anoic acid, and unsaturated fatty acids, they have a suffix with enoic. So the number of carbon atom, it starts mainly from the carboxyl carbon end, and the adjacent carbon atoms are numbered as 1, 2, 3, 4 and so on. Now if you see the structure of fatty acids, they contain a polar head and a non-polar tail. Okay, the polar head is found to be hydrophilic and non-polar tail is found to be hydrophobic. Now coming to the saturated fatty acid, the saturated fatty acids are found to have the general formula CnH2n plus COOH. Now these fatty acids are found to be distributed both in plants and animals. Unsaturated fatty acids, they contain double bond in the structure. Okay, now these unsaturated fatty acids are involved in very important body functions, and they are also found on the structure of cell membranes. Now coming to the main important classification, the first lipid we are going to see is simple lipid. So simple lipid is composed of fats and oils, where you call them as triacylglycerol or triglyceride. All fats and oils, especially the triglycerides, they consist of two main components: glycerol and fatty acid. So you have one glycerol molecule bonded with three fatty acid molecules, and the glycerol and fatty acid are interconnected by ester bonds. So if you see, the main component of simple lipids are the fats and oils. The fats are found to be solid in nature, whereas oils are found to be liquid in nature. Now if you see the properties of triacylglycerols, the triacylglycerides are composed of the first property, hydrolysis of ester. So the hydrolysis of ester reaction is catalyzed by an acid or an enzyme. So the main enzyme which catalyzes the hydrolysis of ester bond is lipase. So lipase is the main enzyme which will catalyze the enzymatic hydrolysis of triacylglycerol to produce free fatty acids and glycerol. So if you see here in the reaction, the triacylglycerols, they are hydrolyzed by the enzyme lipase to produce the free fatty acids and glycerol molecule. Now coming to the next very important property, so all of you know what it is. It is the saponification reaction or the formation of soaps. So this saponification reaction, mainly it is taking place with an alkali, which is sodium hydroxide. So the hydrolysis of triacyl glycerols by alkali, it produces soaps which is called as saponification. So mainly these soaps are long uncharged hydrocarbon tail, and they are mainly involved in the formation of soaps where they contain sodium or potassium salts or fatty acids. Now coming to the next important property called hydrolytic rancidity, where the butters or fats, if they are stored for a long period, they give a very bad odor. So it is mainly due to the enzymes which will convert your fatty acids into glycerol and free fatty acids. Then next important property is peroxidation property, where in living cells the lipids will undergo peroxidation to produce peroxides. Now these act as free radicals and they damage all the tissues, which will form inflammatory responses, aging, cancer and many other heart related diseases like atherosclerosis, and many risky diseases are also obtained since there is increased production of free radicals. Now coming to the next type of lipid, which is waxes. So all of you know what waxes are. So the waxes are made up of simple esters of fatty acids. They are also called as saturated long chain monohydric alcohols. So the two main type of waxes which are seen commercially is called beeswax, where it is composed of alcohol and fatty acid. Then spermate, which is containing cetyl palmitate. Then carnauba wax, which is obtained from the palm tree. Now generally waxes are made up of long chain fatty acid and long chain alcohol. Now coming to the compound lipids. Compound lipid, the first one you call it as phospholipid. They contain the nitrogenous base, fatty acid and a phosphoric acid, and also they contain glycerol, which is combined with inositol or sphingosine. Now these phospholipids are of two types, phosphoglycerides. Under phosphoglycerides you have phosphatidic acid and lecithin. Now lecithin is also called as phosphatidyl choline, where they contain choline in their structure. Then next the phosphoglyceride is going to be cephalin. Cephalin is composed of ethanolamine, hence they are called as phosphatidyl ethanolamine. Then the fourth type of phosphoglyceride is plasmalogen. Plasmalogen, they contain aldehyde group, where the phospholipid is called as phosphatidyl choline, phosphatidyl ethanolamine and phosphatidyl serine. Now coming to the next type of phospholipid is phosphosphingolipids. These phosphosphingolipids are also called as sphingomyelins. A sphingomyelin is composed of three components: sphingosine, fatty acid, phosphoric acid and choline. So all these components make up a sphingomyelin. Now the general phospholipids, they are produced and they are cleaved or hydrolyzed by a specific group of enzymes called phospholipases. So phospholipases are a class of enzymes which will hydrolyze the phospholipids and it will cleave the fatty acids and phosphoric acid present in the sphingomyelin. The third type of phospholipid is the glycolipid. So these glycolipids are found to be the important constituents they are seen on the cell membrane, and they are of two main types, cerebrosides, gangliosides. So the cerebrosides, they contain ceramide, and gangliosides, they are found in all the ganglions in the neuron cells and they are made up of glycosphingolipids. Now coming to the last type of classification is lipoproteins. Now lipoprotein, there are 5 types of lipoproteins: chylomicrons, VLDL, IDL, LDL and high density lipoproteins. Now the third classification is what we are going to see is derived lipids. Under derived lipids we are going to see steroids. So all the non-saponifiable lipids are said to be steroids, where the main type of steroid which we see in our body is cholesterol. So all steroids contain a cyclic carbon nucleus ring-like structure, and this cyclic carbon structure is formed by a compound called cyclopentanoperhydrophenanthrene ring. So this ring is formed in the structure of this cholesterol, where the cyclopentane ring is attached with phenanthrene molecules which form the cholesterol structure. Now all of you know what is cholesterol. Cholesterol is an important steroid hormone. It is found in the cell membrane and it mainly determines the membrane permeability, and it is important precursor for the synthesis of sex hormones and adrenocorticoid hormones. The main property of cholesterol is it is insoluble in water and soluble in organic solvents, and ergosterol and bile acids, they are all the important products of cholesterol metabolism. And next is terpenes. Terpenes are said to be non-saponifiable lipids in plants, and they are found in combination of 2 or more molecules of isoprene units. Then we come to an important property called amphipathic lipids. These amphipathic lipids, they are so called because they have both the polar groups and the non-polar tail. So the polar groups were found to be situated between the aqueous phase, while the non-polar tail or the non-polar groups, they are present in the opposite direction where they form the micelle formation. So micelle formation is mainly formed by the bile salts, where they are essential for lipid digestion and lipid absorption. So now all of you know what is a biological macromolecule or biological membranes. So this slide tells you about the biological membrane. So the biological membrane is made up of 2 layers, hydrophilic layer and hydrophobic layer. So this entire structure of biological membrane is called fluid mosaic model, and it is made up of phospholipids, cholesterol, steroids, glycolipids. Now coming to the last topic of this unit, nucleosides, nucleotides and nucleic acids. Nucleic acids are found to be the final class of biomolecules, where they are called as nucleic acids. Then these nucleic acids are of 2 types, deoxyribonucleic acid and ribonucleic acid. So the central dogma of information, where DNA is first converted to mRNA and mRNA is converted to protein. So DNA to mRNA you call it as transcription, mRNA to protein you call it as translation. Now nucleotide structure, it is made up of 3 subunits, sugar, a heterocyclic nitrogenous base and a phosphate group. So this is the overview of a nucleoside, nucleotide and nucleic acids. So if you see a nucleoside, it consists of only a sugar and a base in their structure. If you see a nucleotide, phosphate is bonded with sugar and sugar is bonded with a nitrogenous base. And if you see the finer nucleic acids, phosphate and sugar and the base, they all align in a structure, in a double helical structure, which forms a nucleic acids. Okay, now we have completed our unit 1 topics. Now we will, in the next presentation, we will see the unit 2 topics. Thank you.